Welcome to the Biotech Talk. I am introducing myself as Molly Zeller, and I am the lab manager for the DNA sequencing facility here at the Biotech Center. I am going to give a talk today on a project I've worked on kind of on the side through our professional society called ABRF, and I'm going to go into a little bit more detail shortly. The title of my talk is it me you're looking for? A multi-platform and cross-site evaluation of technologies and methods used for DNA methylation profiling. I like the title because I like the song by Lionel Richie, and then I rewatched the video for it, and I realized it's a very creepy video. So if you haven't watched it in a while, maybe go and check it out, but I'm warning you. All right, so you'll notice that this is also our 2023 study. We're currently finishing up our 2024 study, which will be presented at this year's meeting, but we are still working through all of the data that we've generated in this, which, as you'll come to find out, is a lot of data. All right, so my professional society that I'm a part of is called ABRF, or the Association of Biomolecular Resources, or Resource Facilities. They're a nonprofit. We're part of FASEB, which is the Federation of American Societies of Experimental Biology. They do a lot of advocacy for us up on the Hill, advocating especially during these challenging times that are unknown to a lot of us right now. ABRF was founded in 1989. Fun fact, it was founded by one of the founders also of the Biotech Center, Ron Neese, and he is a big part of ABRF, and I get to visit with him every year at the annual meeting. We did just pass 3,000 members in ABRF. We span 16 countries, and we cover academia, government, and industry. And a lot of the people that are part of ABRF are core facility people, which I'll show you in just a short minute. This is just my cool little shout out to our annual meeting that we're holding this year. It's in Vegas, and I really liked our sign, and I liked our little slogan that says, "What happens in cores stays in cores." So everybody will be convening in Vegas on the Strip in about a month to discuss everything that happens in core facilities. And I am extremely looking forward to it. All right. So as I said before, ABRF has a bunch of different members. A lot of them are directors, staff scientists, managers, and administrators. So anybody who's kind of involved in a core facility is welcome to join. And we have different pathways to communicate, to collaborate, and kind of, learn and how we can navigate the world of core facilities, and especially on university campuses. And then also, ABRF is very heavily dominated, I would say, by genomics core facilities. So if anybody is listening, or if you know people who are not part of a genomics core facility, and they're really wanting to join a professional society, I would highly recommend you send them our way. You can direct them to me. I'm probably ABRF's biggest fangirl, so happy to help get them signed up. All right, a couple more fun facts about ABRF, just because, like I said, I am a very big fangirl. We do have some regional chapters, and the reason I bring this up is because I don't know if anybody recognizes this beautiful city. Anybody? It's Madison. Guess what? The annual chapter meeting is going to be held here this fall. So if you're going to be on campus and you want to learn more about core facilities and the challenges we face with, projects, campus, universities, money, government, whatever, feel free to sign up. And registration opens up in July. Once again, contact me. I'm happy to help. I am organizing it. But October 22nd through the 24th. All right. So, like I said, the ABRF has a lot of opportunities for members to kind of participate in different kind of committees, and we collaborate with all sorts of kind of things. We have like flow cytometry groups, we have DNA sequencing groups, we have membership committees. We have corporate sponsors that like we collaborate with as well. And I'm part of the DNA sequencing research group. As you can see, I'm also the co-chair this year. I will be chair next year, and we are kind of a conglomerate of people from all over the United States. I mean, I put where everybody's from kind of after their name there. So Florida, New York, Indiana, Michigan, all sorts of places. We meet every other week, so these are probably some of my closest friends. I think I talk to them more than I talk to my like real life in-person friends. But we're all really work together. All right, this study that I'm going to be kind of talking to you about the results today, our methylation study, didn't just take the DSRG. We had to pull in help from everybody that like we knew. Like you can see I have two people from the University of Wisconsin-Madison that helped, Emily and Sandra. And Emily, I would probably say she is the one who organized all of the DNA for this project, which once I get into explaining the project, you'll understand how much DNA we actually had to aliquot and shear and QC and prep. And she did all of that, but she is now in grad school and working on amazing things. So, and not here, unfortunately. But yeah, just so you can see, and then all the people in green are actually all the people that we had to enlist to help with bioinformatics. So the DNA sequencing research group is a lot of people who do DNA sequencing, but don't necessarily analyze the DNA sequencing. So we had to enlist people from Cold Spring Harbor and WashU and UT Austin to help us analyze all this data. And we actually still meet regularly with them because we're still digging through this data. All right, so in terms of costing like projects out, and if you are part of a core facility or you understand kind of how this works, there's not just the reagents, there's labor, there's consumables, there's the consultation, there's the design time. These are the sponsors that we have worked with for this project. Now, we rely on vendor donations. We can ask for a small budget from ABRF, but think like $5,000 for like shipping costs and maybe hiring, a consultant or something. Does anybody want to take a guess? In terms of vendor reagents, how much do you think a project, and I know you don't know the specifics of a project like this yet, I haven't quite introduced it, but if you can just guess ballpark of doing methylation across 3 different platforms, what that would cost. We're talking human samples too, by the way. Million? That would be great. We did not quite get $1 million from vendors though. However, we did get, and it's very small at the bottom of the screen there, but it's $103,000 that we got donated to us. And I actually was just going through this beforehand, and it's actually more like 110 because we did have to get another 10B flow cell from Illumina to kind of finish out some sequencing. So we're looking at about $110,000 just donated to us. So all of the time that is spent putting in, to these projects, that time is volunteered by us. So that's something that I work on the side. And we, take this project, like I can do a lot of things at home or stay late after work and shear some DNA on the side. But you might think, well, what do vendors get out of all of this? Why would they donate all these reagents? It's not like they're just like, hey, we like you, we're going to give you free reagents. They actually get a lot of say in this project. So we collaborate with them before we start doing the work on how the best to handle their reagents, what samples they want us to use. And they also get a lot of like marketing from us. So here, I'm marketing for them. Like I'm explaining to you why we've used these products, what the benefits are from them. So these vendors do get a lot of kind of airtime, if you will. And we do also give them the data that we generate from their own technology. So like PacBio gets the data that we generated from the PacBio technology, et cetera. All right, so you're like, all right, Molly, let's get into the science. That's why you're probably here, right? You don't want to hear me talk about ABRF, even though I could talk about ABRF probably for, I don't know, 2 more hours. All right, so this is kind of our design of our project. And it's a little bit busy. This was originally our slide kind of for our proposal, so it's not probably the best for a presentation, but I wanted to kind of put it up to show you kind of how large this project really was. So we started with 4 DNA samples. We wanted to do everything in human because human is, you know, basically what everything gets tested in. So we have Na12878, we have a breast cancer sample, and then we have 2 methylation standards. One that is supposedly 100% methylated and one that is supposedly completely unmethylated. You'll find out that it's hard to be completely or completely not methylated. But I'll get into that shortly. All right, so then we decided to do whole genome sequencing using short reads and long reads. So we kind of had to split it out because in a different kind of, a little bit different paths. So you can see in short reads, we wanted to check out bisulfite conversion, but we had 2 different vendors that wanted to participate in this. IDT wanted to participate with their bisulfite conversion kit, and Zymo wanted to participate as well. And then we also wanted to try out NEB's new enzymatic methylation kit as well. So that's what we did on short reads. And we actually sequenced them on 2 10B flow cells. All the sequencing was done actually at Washington University in St. Louis. And then for long reads, we don't have to do any conversion there. So we're going to sequence the native DNA. And then we did that on the PacBio Revio and then the Oxford Nanopore Promethean. Those were also done at WashU in St. Louis. All the sequencing was done there. All right, so just once again to touch briefly on the samples, we used 12878, which is the human cell line. And then we have MCF7, which is a breast cancer human sample, and then our 2 DNA methylation controls, both from Zymo. So I mentioned my colleague Emily, who had to kind of do all of this. So not only did she have to quantify, like, I think we worked with like 30, 40 micrograms of DNA to get this project started. She actually had to go and shear acoustically using Covaris shearing, mechanically using kind of G-tubes, also from Covaris, and then also some DNA didn't go through any shearing. So we have 3 paths for the DNA to go through there. And then once we had the shearing done, we had to convert the DNA using bisulfite conversion. That DNA, you know, either some were sheared and then also had to go through chemical conversion or enzymatic conversion or no conversion. So this is like kind of untangling like a web of how do we get all this DNA where? And not only was this just happening in one site, we did this across 3 different sites for each prep. But not just 3 prep, not like site A, site B, site C. We had site 1, 2, 3 for the Zymo prep. We had site 1, 2, 3 for the NEB prep. So we're talking, I think across 12 or 13 different sites total, we had to send different aliquots of DNA, sheared, converted, everything across the country. All right, and then I think I also forgot to mention that we also kind of wanted to compare this to the gold standard of methylation array, which is the EPIC array from Illumina. So we also threw that in as well. Okay, so this is just kind of show that we did do our due diligence and check that our DNA was of good quality before beginning. We started with some high molecular weight DNA that we extracted from cells, and then we sheared. That second graph here, we sheared for long read sequencing. I think it's, about, I can't even read that number, like 15 kb, I think. And then we sheared for short read to about 350 base pairs. All right, so this is a little side note here. We took some surveys across the people who actually had to do the prep. So like I said, we had a couple different sites doing each prep. And you can see here for this first graph, the overall and there's a typo, overall ease of use for each kit. And you can kind of see that a lot of people thought it was relatively, easy, but also, I don't know, I feel like it's a little bit not the same across. So like, just think like we've got people who might be, early techs who are doing this versus people who have been working in the field for a while. And then the second graph here, was the protocol easy to follow? Also can kind of see some varying opinions, but I would say on a scale of 1 to 5, everybody was at least between a 1 and a 2, so not too bad there. All right, this graph kind of makes me chuckle a little bit. You can see the total hands-on time here, especially looking at the Zymo Illumina kit. It apparently only took one person 1 hour of hands-on time, but it took another person 4 hours of hands-on time. So either somebody doesn't know how to track their time very well, or they're skipping steps, I guess. I'm not 100% sure. So like I said, this was just a really quick survey we took of the users that we had prepare in our project. Okay, all right, so enough of the background. However, I'm just going to touch a little bit on what is DNA methylation. So we've got epigenetics versus genetics, and I'm sure everybody who's either listening virtually or is in the room could probably explain this, but I'm going to do my due diligence and just touch on a few points. So we've got genetics, which we're going to, the heritable changes of the gene activity due to like direct alteration of the DNA, such as point mutations, deletions, insertions, translocations. In contrast, epigenetics is going to be the study of, the heritable changes in gene activity that is not associated with the DNA sequence in and of itself. So it's kind of those modifications that are made to the DNA. You can kind of see those methylation groups on the top of the DNA strand shown on the right. So why is the DSRG interested in this? So we are the DNA sequencing research group. So one could say, well, it's a DNA sequencing methodology, so that's why the DNA sequencing research group is interested. Yes. However, I'm going to go back to ABRF. So we're a core facility. So we have services that we need to provide to our clients, right? And DNA methylation is one of those services that clients are interested in. They want to know what, they are studying those epigenetics, but they need a way to do it that's not epic array, or there's, they have maybe non-model organisms, so they can't use an epic array, or, they want to study across a multitude of species. So we wanted to do this because we want to help our clients. We want to be prepared and have the knowledge that we can share with our clients when they come to us and asking for this service. All right, so very briefly, how does methylation get detected in short reads? So there are two methods, as I kind of briefly touched on earlier. We have the bisulfite conversion and we have the enzymatic conversion. So bisulfite conversion uses sodium bisulfite to deaminate the cytosines into uracils. And then those uracils, the cytosines that are not methylated, and then those uracils get translated to, or transcribed to Ts during PCR later. And then when we go to sequencing, all of the Cs that get sequenced, those are the methylated Cs in the sequence. And we have enzymatic conversion, which theoretically does the same thing, just with a different method. So they're going to use TET2 and ApoBec. And gold star to anybody who can tell me what ApoBec means or stands for. I can't see chat, so sorry if somebody in the chat is like really like, oh my God, I know what this is. But anybody here know what ApoBec stands for? Trust me, I looked it up. It's apolipoprotein B mRNA editing enzyme subunit 3A. So fun party fact later, right? Okay. All right, so that's short read methylation detection. You actually have to modify your sequence in order, or your DNA in order to detect it. However, we look at PacBio and Oxford Nanopore sequencing, we're actually sequencing the native DNA. So PacBio is actually going to use polymerase kinetics. So in this image down at the bottom left here, you can kind of see on the top is kind of a normal reading of G, A, T, and C. And how they determine that is they measure, the time that it takes to read those bases. However, when a methylated cytosine comes along, it takes a little bit longer for it to go through, or for the polymerase to read it. And so they can use that time to calculate and do all fun things and tell us that is a methylated C. So we're reading the direct methylation from that native DNA. No modifications, no conversion. It is what it is, kind of thing. All right, so Oxford nanopore does a similar thing, although it's not a polymerase. What they're actually going to do is they're going to check the current, or kind of use the current change through the nanopore to determine what that base is. So they didn't have a cool ACGT thing like PacBio did, but as you can see, as on the right there, as the current changes over time, or the current signal changes over time, when it hits a methylated base, which I think they're showing as the red little dot, I guess, that the current changes, and they can tell that that is also a methylated base. Okay, so you could probably say, Molly, I've read papers about this. This is not new information, right? Other people have done studies on this, have done a lot of comparisons on the different kind of epigenetic sequencing techniques. However, we wanted to be pretty thorough, include more technologies. We wanted to add some interesting samples. And we also wanted to provide the transparent processing methods. We wanted to be very transparent with our clients, with the vendors. This wasn't just for us to get another paper under our belt. We're doing this so that we can share this information with our clients, which is why I'm here sharing this information with you. And then we also wanted to, when we do publish, we're going to publish it more as kind of a guidance, like how would we present this service to our clients? What would we want to do? All right, so I'm going to give a quick little forewarning or disclaimer. I'm not a bioinformatician, so I am going to go through some graphs, and I might not have all of the answers because all these graphs do come from our bioinformaticians that I mentioned earlier. So there's my disclaimer. All right, so I'm going to start with some Illumina QC metrics that we did, that we got from the NovaSeq X Plus in WashU. So on the left here, you can see we have our sizes. So the top, the bluish purple box are going to be the bioanalyzer library size traces for each kit. So we have IDT, NEB, and Zymo. And then the pink is going to be the actual insert length average. That's going to be the size of the read post adapter trimming, post filtering, and everything. So you can see they're obviously much smaller, but they do have the same trends as their bioanalyzer traces. And then we were just kind of graphing, trying to see how well the library sequenced, and we did pool them by the same molarity on the flow cell. However, it looks like Zymo, we did end up getting probably about almost twice the number of reads that we got on the flow cell. Sometimes this happens. As a person who does a lot of Illumina sequencing, I can tell you that some libraries just sequence a lot better than others. All right, so then we have just looking at the kind of the Q30 scores for each kit, still on Illumina sequencing. We have really good Q30 scores. Everything's above 90% for each kit, variable across read 1 and read 2, especially for NEB. And then we do have percent duplicate reads as well on the right. Okay, so one QC metric that we do to kind of test for methylation is the mapping efficiency. So we're looking for the mapping efficiency for the cytosine methylation calling. And as you can see here, IDT and NEB have increased better, I guess, I don't know if you can say, mapping efficiency when it comes for the cytosine methylation calling. Now, do we know why? that's something that we're still looking at the data for. Like I said, we have a lot of data, so much data. Uh, it's going to take us a while before we get through everything. All right, so now we're also going to look at the conversion rate for the libraries from NEB and IDT and Zymo. So you can kind of see that the conversion rate was all pretty similar across all three kits. Not too many differences there. And then we also looked at the Lambda control as well, because the Lambda control gets included in all of the library preps. Okay, good correlation across all three samples. And then once again, showing the conversion rate. And this might be like, oh my God, IDT is much different from the rest, but let me show you the scale here. We're still looking from 95 to 100%. So we're all pretty much above 95% for all the conversion rates. All right, so I'm going to take a step away from Illumina for a sec, and we're going to move into some PacBio QC that we got from WashU. So each library here, we got about 10 million reads. Our read length was about a little over 10 KB long. So remember, we're thinking Illumina, we did 2x150 sequencing. Here we're taking 10 KB read lengths. So we've got quite a bit difference there. And then all of the samples did generate around 30x coverage. So this is also just kind of showing that we can be very consistent. All the libraries were prepped at different sites, but all sequenced at WashU. So this just kind of shows the consistency and the reliability of PacBio sequencing and library prep. Okay, so once again with ONT, libraries were prepped at different sites. They were actually prepped in duplicate and all sequenced once again at WashU. This is not an advertisement for WashU, I promise. And so this is just kind of showing the yield that we got across all the samples and our read length as well. So you can see yeah. So we got our yield are the bars, and it looks like we got 100 gigs for most samples, I would say. Okay. And then the next slide is actually going to show the coverage. So once again, about 25, 30x coverage for all the samples from ONT. So you can tell from all of these samples, we did reach about 30x coverage for everything. And that was also including Illumina. I just don't think I don't think I have a coverage slide on the Illumina QC. We are currently working on downsampling to 20x, 10x, and 5x across everything to kind of see, what level we can use or that we need for methylation calling. Okay, so we did also do the epic array, as I mentioned. And once again, we did 3 different sites. These 3 I actually have shown here. So we did Dartmouth, Northwestern, and here, UW. And you can kind of see that the largest variation is going to be across sample type and not site. So once again, there is reproducibility in these protocols, in these assays, which is very good to know, and obviously good for the vendors to know that, they can give their kit away and it'll be good in any hands. And these samples are I would say pretty different when it comes to, we have one kind of normal human, one breast cancer human, and then we've got 2 methylation or methylated, unmethylated samples. All right, and this is showing across method. So this is showing kind of across the method how reproducible everything is together. So and I know that the symbols are all very much jumbled together, but promise me I can tell you that they're all the different methods. So Hi-Fi, IDT, NEB, ONT, Zymo, everything is kind of all grouped together for each sample type. So the colors are the different sample types. So the purple is the non-methylated, green is methylated, The reddish pink is our MCF7 sample, and then our blue is the Na12878. And I know we have one outlier. That was one of our PacBio library preps, just slightly off, but I would not consider that to be too terribly off kilter, but does kind of make for a interesting question about the graph. Okay, so once again, I'm going to bring up WashU again. They have some amazing scientists there, and we actually collaborated with the Wang lab from WashU. And what they have is they have this browser. It's an interactive data hub where you can basically, like, I can look across the whole genome and like look at these tracks. So the tracks here, we have our gene annotation, the repeat annotation, and then what we're really interested in for our project is our methyl C tracks. So in each kind of track here, we have our read depth is going to be that black squiggly line that goes across. And our methylation, our percent methylation is going to be the blue shading, which this track is specifically, these bottom three are showing you some very high methylation coverage right there, okay? And I'm going to show you some more examples. So I just wanted to kind of point out what we're going to be looking at in the next few slides. All right, so this shot is going to show how reproducible the replicates are within the sites. So these are across our different sites. This is actually using just the Zymo Illumina kit. And you can kind of see across. We have our methylated sample on top, our non-methylated samples at the bottom, and then our two human samples are in the middle. And then our sites, their replicates, are next to each other. So you can kind of see across all sites, especially for that methylated, I mean, nothing comes to my at least untrained eye right now, you know, looking at that and says, oh my God, there's some glaring differences. And everything looks the same for MCF7 and Na12878, and also the non-methylated samples. And here's also where I wanted to point out that I said before that our methylated sample was 100% methylated. You can kind of see, I mean, it's not super easy to tell on this picture, but there are some little dips where you can tell there is some less methylation, some fewer methylated sites. And then also for the non-methylated sample, we do have some areas where there is methylation present. But like I said, it's hard to completely remove all of the methylation from the DNA. So once again, this is showing the replication between the sites. Now I'm going to show you the reproducibility between methods. So once again, slight different order of our samples here, but we're going to show ONT, so the Oxford Nanopore, our PacBio, IDT, NEB, and Zymo, all for our methylated sample. Up at the top, once again, these patterns look very similar across the different sample types and through across the different methods. So one thing I've found really interesting just going across this is in this data hub, you can actually like zoom in, and if you have like a favorite gene you want to look at, I can zoom in on that gene, check out, you know, where there's methylation and where there's no methylation. And even here, you can kind of see the differences right away between the normal human sample, the 12878, and our MCF7, kind of in the middle there. We can see a region of no methylation in the normal sample, but there's a region of higher methylation in the cancer sample. So one example of how the two different human samples differ. Okay, so another fun thing about this data hub is we can kind of look at and kind of maybe figure out, are there any discrepancies from the short read sequencing to the long read sequencing? And like I said, so the long read sequencing, like I said, is native DNA. So we're kind of like looking at the methylation as it stands, as we put it onto the instrument. For the Illumina sequencing, we actually have to manipulate that DNA. So what if it's not 100%, right? We can see here where our hi-fi sample, which is the top track there, identifies methylation a little bit further on down, but doesn't find it in our Illumina short read sequencing. So we can kind of find an instance where maybe the Illumina sequencing didn't catch it, or the bisulfite conversion didn't, completely convert everything over. And then we have our Illumina sequencing here on the bottom, or the Illumina short read, and we notice that we have some coverage or some methylation sites there, which could be maybe spurious sites that are incorrect. So these are also one of the other things that we want to look at with our data set, is to kind of identify where we can find these different sites, and if there's any corrections that need to be made, or anything that's very interesting throughout maybe somebody's favorite gene. Okay, all right. So then we also can kind of correctly characterize methylation patterns of the SNVs using long reads. So like I said before, in bisulfite conversion, every time you read a C, that's going to be the methylated site, which that means when we're reading Ts, that is the unmethylated cytosines, or it's just Ts. So we can see here, Hi-Fi said, nope, there's no methylation, which also correctly confirms on our short read sequencing on the right there that there is no methylation, because that site was also called as T. So we can also use this as kind of a confirmation to make sure that what we're seeing in the short read is in fact correct. All right. So in the end, all 5 methods can reliably characterize the CpG methylation in our human cell lines. We found partial methylation of our Zymo standard non-methylated samples. And the third gen sequencing reveals spurious methylation calling from the bisulfite or enzymatic methylation sequencing that I showed in that last, or second to last slide. So I know I presented a lot of slides to you today, and we're barely scraping the surface of our data. We still have so many things that we want to look at. One, we're going to integrate the EPIC array data into all the other data that we have here. As I mentioned, we're working on downsampling, which I think we have finished, or we're almost finished with. And then we're going to then phase and look at allele-specific methylation detection. So this is where we're going to go through. We'll look at, those favorite genes, those areas of concern, and go across. And then we also want to look at, the difficult to map regions. And then, just looking at any kind of calling differences that we have between these methods. So once again, working with our amazing bioinformaticians from Cold Spring Harbor, UT Austin, and WashU. We use a lot of WashU's compute to do a lot of this data analysis. And honestly, I work with an amazing team, and this is a really fun little side project that I get to work on. And I'm always happy to talk about it. So stay tuned, because this is not the last you're going to hear of me on this project. This is kind of the beginning. So yeah, so with that, that's all I have for today. Just because, like I said, we're at the very beginning. Does anybody have any questions on this? Ooh, how do I expect to integrate the array data? Leah, that is a question for a bioinformatician. And I gave you my disclaimer, I'm not a bioinformatician. That's a question for my friend, actually at UT Austin. That's our array people. So I can get back to you. Yes, we have. I don't have those slides, but we have looked at it. Things actually match up quite well. So yeah. So I can try to answer questions. What's up, Dustin? Yeah, they did after we literally we got through all of the kind of the calls, the downsampling, looking at the coverage, and I got an email from our ONT person like, hey, we just made some updates. And I was like, cool. So that'll be in the future. Yeah, I'm sure we'll get somebody to do it. But yeah, we are still on the old. So everything's, things move so fast now. Yeah. Uh, Bismarck. Is that the right answer? I think that's the correct. I know there's tools. I just don't know which one goes to which, but Bismarck, I think, is the answer for you. Any other questions? Question. Oh, yeah. That is a great question, and it is something that we did notice kind of in the early QC is that um, we did see the difference in the conversion rate for Zymo. I think you could see that was a little bit less um, efficient, I guess I should say. Do I know the reason for the differences in the directionality? No, I personally don't. I actually just asked my boss about this before, and we both were like, we're going to have to ask somebody who knows a little bit more about the directionality. So I'm going to respectfully say I don't know the answer to that question. But I do think we are looking into it, and the people who, my team is going to answer that question eventually. So, any questions about, I don't know, I mean, methylation, or also, like I said, I'll talk about ABRF for days. So, what's up, Dustin? Nope, this one. Oop, ah, yeah, absolutely. That is a really good point. Just kind of summarize for people who can't maybe hear you on the call. Just talking about how in this slide how there are the repeat regions, and that's where a lot of the methylation happens. And if short read can't pick it up, it just shows the power of our long read sequencing and why native DNA sequencing is I was going to use the word superior, but I don't think I should use that word. But it has a lot of benefits, especially when it comes to looking at these epigenetics, right? So I think what you said, Dustin, is very important. That yes, these different regions are a really good spot to look for these different differences between the methods. Yeah, correct. Yeah, yeah, me too. Yeah, we're presenting again this year in Vegas, and I think we'll probably start working on a paper after that. So we actually our last year study was a big single cell study, or actually 2022's was a big single cell study. So we're just finishing up that paper, if that tells you the timeline of our stuff. So this year's study, since our last two studies were massive undertakings and we're still digging through data, we decided to do a smaller study of just kind of surveying fellow cores on kind of wet lab practices, number of clients, and kind of how they use like their admin practices and stuff. Just kind of like a survey that we're going to present in Vegas. So we took it a little light this year, but we don't have room to store more data. So Anybody else? All right, well, I think we can be done. I don't know how to end this. Thanks. Okay, thanks.